Hello, my name is Jason Linville. This is CJ352, just another mini lecture on amplification method. What does that mean? What we're going to do in this shorter video is I want you to be able to list the components of the PCR reaction. Well, wait a minute, can't I already do that, Dr. Linville? Isn't that the only thing you want us to know from the PCR lecture? You're correct. But now I just want you to be a little bit more specific for if we're amplifying STRs. Okay, what do we put into the tube? So I'm just going to ask you to describe the primers, DNA, polymerase, DNTPs, maybe a little bit more closely. Okay, calculate the number of primers needed given the number of loci. So I'm going to say, okay, you're setting up a reaction where you're amplifying 5 loci. How many types of primers go in the tube? You're going to have to know the answer to that. It's 10. Okay, then you're going to calculate the number of alleles amplified given the number of loci. So I'm saying you're going to amplify 7 loci in this reaction. How many alleles will you wind up observing after the reaction is over? And you're going to have to give me the answer to that. It's going to be 14. It's times 2. There, I saved this all to 10 minutes. No, but let's actually look at why it's times 2 for both of those. Okay. So the way, and this leads into the lab that we're going to observe next week, or would have observed, or I don't exactly know how to phrase that. But what we're going to do is, I want you to sort of visualize that we're setting up an amplification reaction. Okay, we know that there's 2 things that, or 4 things that go into the tube, but we want to start visualizing, okay, if we're amplifying STRs, what exactly is on the bench in front of me? Okay, and what exactly am I squirting into the tube? So we're going to start with, let's say we're just amplifying one of those locations of DNA. Okay, so we're just going to amplify one of them. When we analyze the amplified DNA, it's just going to be 2 alleles that are amplified. Remember, one location, everybody has 2 alleles, giving us a pair of numbers. Okay, so how do we set up that reaction if we just wanted to amplify D5S818? Okay, well, number one, we need to recognize that this is going to come from a person, right? So let's even get more specific. We're going to say we're going to amplify D5S818 from Jason Linville. So how do we set up that reaction? Well, number one, in all these reactions we're going to talk about, the polymerase is the same. It's Taq polymerase that we talked about on Wednesday of last week, or in a couple lectures ago, right? The DNTPs, the AGCs, they're going to be exactly the same. It doesn't matter what area of DNA you're amplifying. Those 2 things are going to be exactly the same. The DNA, here it's my DNA, but if we want to amplify like a disease gene in my DNA, okay, then it would still be my DNA. So my DNA, or the person's DNA, it's going to change depending on who you're trying to get the profile from. So when we go from just generic PCR to amplifying D5S818, I want you guys to recognize the only thing we need to change are the primers, right? Because the primers, the sequence of the primers are what controls the area of the DNA that those primers attach. Okay, so if we're amplifying D5S818, we need to choose primers to amplify D5S818. Okay, we already covered this a little bit with PCR, but this is to reiterate. If you want to amplify D5S818, you need to choose a forward primer. Okay, so let's say this is the area of the twisted ladder and we've untwisted it. And D5, there's a whole bunch of bases in here. So we want to amplify this area of DNA. Okay, we need a primer that attaches right here, the forward primer on one side of D5S818, and a reverse primer that attaches over here on the other side. Okay. So one location that we're amplifying, we need 2 primers, a forward and reverse, and then it amplifies what's in between it. Okay. Now, we need 2 primers to amplify D5S818, but also we're going to get 2 alleles when we amplify D5S818. But I want you to recognize that it's always times 2, but it's not as though the forward primer is amplifying one of the alleles and the reverse primer is amplifying the other allele, because you can hear, oh, 2 primers equals 2 alleles. 1 primer is for one allele, one's for the other. No, no, no. It's a forward and reverse primer, and the forward and reverse primer is going to amplify this allele, and the forward and reverse primer are going to amplify this other allele. Okay? So 2 primers for D5S818. Now this is sort of cheating because the primer is like only 3 here, okay, and it matches because remember, G's go with C's and C's go with G's, all right? So this is going to sort of, this primer that we order is going to line up here. Now in reality, this area of DNA is probably, primers are probably about 15 to 25 base pairs long. And you can imagine if you have a primer that's 15 to 25 base pairs long, it's not going to attach anywhere in the DNA. It's going to be looking for that specific sequence in your entire 3 billion letters, okay? Now, for one given primer that's 15 letters long, we want it to attach right here, correct? This primer, and we'll call this the reverse primer, and we'll call this the forward primer over here. This reverse primer, it's probably going to attach somewhere else in the genome, but amplification is only going to be successful if a reverse primer attaches a certain distance, you know, within 50 to 1,000 base pairs from the other forward primer, okay? Then amplification will occur. So when you choose 2 primers that are 15 base pairs long, okay, and the commercial companies actually do the research and development to make sure this is choose, they are only going to attach next to each other at one location in the genome, okay? And that location is going to be D5S818. So those are only going to attach at that location in the genome, and when amplification occurs, you're only going to amplify that one location, okay? So again, one forward, one reverse primer, okay? They're both going to bind on the chromosomes of one's going to be upstream of the location, one's going to be downstream of the location. Okay, and since there's 2 chromosomes, 2 copies of that location in the human genome, you're going to wind up with 2 alleles. Okay, one location equals 2 primers, forward and reverse. One location equals 2 alleles, one from your mother, one from your father. Okay, so just sort of try to get those straight in your head. Okay, let's change it up a little bit. Let's say we want to amplify 3 STR locations. Okay, from Jason Linville. And this is called a multiplex reaction. It just means that it's not a we're not amplifying one area of DNA, we're amplifying several at the same time. And the challenges with multiplex reactions, and the reason why commercial companies have to put so much research and development into developing a reaction that can amplify, 13 loci at one time, is that if you have a pair of forward and reverse primers that work better than another pair of forward and reverse primers, that amplification of one location tends to consume all of the Taq polymerase and all of the DNTPs, while the other one sort of falls behind. And we know from amplification, sort of taking off as far as that exponential curve, it's very important that all of your loci sort of take off at the same time. Other one's going to get left behind and not amplify, okay? So with one location, you don't have to worry about that. But with 3, you start to have to worry about, choosing your primers carefully. Again, when you're amplifying, setting this up for different reactions, amplifying different things, the only thing you have to change is your primers. You don't need to use some multiplex polymerase you don't need to use special DNTPs, or you have to use a special extraction of Dr. Linville's DNA. No, this is all about choosing your primers so they work together, okay? So for 3 SDR loci, what do we need to put into the tube? Whoops. Oh, I'm sorry. I can't see the number here. I'm already on 13. We're going to go over 3 later. So if you want to amplify 13 SDR loci, what do you need to put into the tube? And this is why commercial companies get paid the big bucks. You're going to put in 13 forward primers, 13 reverse primers. So how many is 13 plus 13? 26. Okay. How many alleles are going to result? 26. Why? Because it's 2 alleles at each of the 13. All right. So again, I mean, this is sort of easy once you know how to do it, but I don't want you guys to get confused. It's just the number of loci. Okay. You double the primers and you double the alleles. Okay. So it's going to be pretty much the same exact thing. Each primer set binds on both chromosomes. 2 chromosomes for each of the 13, results in 26 alleles. All right. And these primers, I mean, you order these primers from a company. So you basically just order from a company like Opera and Up in Huntsville does this, and you just type in like your 15 letters and say, hey, send me a primer, and they send you that primer. It's not just one. It's like millions and millions of copies of that primer that's in the tube that you use. But then you add water and that becomes your primer. Okay, so let's say, I swear I thought I took Dom DeLuise out of these slides. Let's say we're sitting down and we go, okay, we're going to amplify D5S818 from Dr. Linville. What do we need? DNA. Okay, well, that's going to be my extraction. If we were amplifying your DNA, we would take your extraction that you did in class when you swabbed your mouth and then put it through all the steps. Take that out of the freezer. It's in a tube in front of us. Okay, primer 1 for D5S. We've ordered that from a company. So it has D5S and we've diluted it to the right concentration. Boom, 2 tubes on the table. Primer 2, we've ordered it from a company. Boom, that's another tube. Taq polymerase, that's a tube that we've ordered from a company. We get it out of the freezer, stick it in our tray, Taq polymerase. DNTPs, we've ordered that from a company, okay? And the Taq and the DNTPs that we use for all these reactions is the same. Put it on the tray. We need to make sure our reaction has the right concentration of, or is at the right pH. It has enough magnesium chloride for our reaction to work. So we get what's called a reaction buffer out of the freezer. Okay, this is our reaction buffer. It contains all these things that I said you guys don't need to know or memorize. And oftentimes with PCR, you guys don't need to know this. They add the magnesium chloride in a separate tube because sometimes for amplifications, you want to add a little bit more magnesium chloride and sometimes you want to add less. So just because the magnesium chloride is something that the concentration can affect the efficiency of the reaction, sometimes that comes in a separate tube. Okay, so one way to set up this reaction is to get out 7 tubes of stuff, calculate how much of each of those things we need into our reaction, and then it's just squirt, 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 squirt. Was that 7 times? Hopefully. And it's in there. Okay. If we were to do this in our class, this is what we would do. We would not get out 7 tubes because for whatever reaction you do, what did I just get done telling you? The Taq's going to be the same. The DNTPs are the same. Okay, if you're using just a normal amount, the traditional amount of magnesium chloride, that's the same. They actually many companies sell this material and it's called a PCR mix. It has everything you need to do the polymerase chain reaction in it. The only thing it doesn't have is the DNA. You provide that from your suspect. And the only thing it doesn't have is the primers. What area do you want to amplify? Well, you pick your primers and use this PCR mix. So you can use this PCR mix, pull it out of the freezer, pull the tube, and use it for whatever DNA amplification you want, okay? So this is what we would do in our class if we wanted to amplify D5S818. 4 tubes out on the counter. All right, and this is what our lab's going to be about, where I want you guys to understand what the tubes look like and what's in each tube and how to calculate how much we need to put in the reaction. So we'll do that next week, okay? PCR components, okay? Let's say that we're amplifying not 1 loci, but we're amplifying 13. Well, we're going to buy it from a company, right? If you buy a kit from a company, what do you get? Well, you don't get the DNA, right? You need to provide the DNA from your suspect, okay? So you provide that. But what comes in the kit is a tube of primers, okay? And they have balanced these 16 sets of primer, 32 primers. They have them in the correct concentrations. They're not all the same concentrations, okay, to where every loci is going to be able to compete and all of the amplification is going to occur across all of those loci. All right. They also give you a PCR mix, everything you need to do PCR, except similar to before, in these kits, they tend to keep the Taq polymerase separate. And the reason is sometimes when you're dealing with degraded DNA in a forensic science lab or inhibition, which we'll talk about later, you want to give it a little bit extra tack. So the reason the commercial companies don't put the tack in with their PCR mix is they want to give the forensic scientists freedom to increase that amount of tack if needed. But typically the amount of tack per tube is sort of like, I think it's like 0.2 microliters or something. And, you know, you use the standard recommended amount of tack polymerase. But it's still, you're getting out a certain amount of tubes, not 1,000 tubes, okay? Putting them in front of you and then just squirt, 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 squirt in the tube, close it, and you're done, okay? Now, if we were to do this in our class, we're going to amplify more than one, but we're not going to use the expensive commercial kit that we only have a little bit left and it's probably expired. We actually, for our research and teaching in our laboratory, we actually have created our own multiplex reaction. The reason I chose 3 is we tried to do 4 and it didn't work. But if we look at 3 regions for everybody in a class of 24, everybody tends to have, if you do the statistics where it's like your profile is going to match 1 in 1,000 people, everybody's going to have a different profile. Okay, so it's good for when we run all of our DNA, if we were allowed on campus to do that, we would see that everybody would have, okay, 3 pairs of numbers, and everybody would have a unique 3 pairs of numbers. Like your profile, your 3 pairs of numbers would be different from everybody else's in a class. So it's really good for teaching and research. Okay, how do we do it? Well, we get the DNA out of the tube that we did in class, just like we do for everything else, okay? We make up this tube of primers by ordering 6 different primers and then squirting them in the correct concentrations in the tube. We are extremely lazy. We found the same concentrations for all primers, works fine, okay? But we're not dealing with 13 loci, it's just 3. And then we buy the aforementioned PCR mix that has everything we need for the reaction. So if we were to do this in class, you would have 2 tubes in front of you, primer tube, a PCR mix tube, and then you'd also have a tube of water to get it at the right concentration. And then you'd have your 4 extractions that you did. We would make up these amplifications, and that's what I'm going to demonstrate next week for you. Okay, we're going to hold off and talk about statistics next week, and then I'll describe the assignment that you guys are going to do for that. But right now I'm going to leave this recording running a little bit for those watching this video in the spring of 2020 in case you guys have any questions about the class that I forgot to cover or mention. And then I'm going to stop the recording and then you guys can ask me any other questions. Okay. But for now, if you're here and you need to go, now's the time to get out of here. If you have questions, stick around and I will answer them. Thank you, everybody. All right, are there any questions about the class that we want to have recorded that might benefit somebody else who's watching this later? And also, I'm interested in, and you guys can type it in the chat, or if you guys want to turn on your microphone, you can do that as well. Yes. What is slides? Do I have slide numbers on my slide? What is slide 17, Elise? Can you tell me what is on it?
Can you hear me?
Yes, I can.
Okay. It's the one back when we were talking about the locus, the loci and allele, where it talks about each person having 2 alleles, and most of the time they won't have the same. I didn't really understand what you were saying about the 1, 1, the 2, 2, that one. Yeah.
Okay. All right. So this like person 1 and person 2. Okay. So what this slide is supposed to be showing, Elise, is like person 1, if you zoom in on whatever the locus happens to be, you're going to find that in their body, what the pink is sort of trying to represent, where's my pointer? What the pink is trying to represent is sort of like, this is the chromosome that you have from your mother. So if we were to zoom in on that chromosome, we would find that that chromosome is like a series of letters that is exactly the same as the chromosome that you receive from your father. And after the STR, it's exactly the same on your mother as from your father. And after this STR, it's exactly the same on. So in other words, the sequences before these two dots on all three people on all of these chromosomes, it's exactly the same in all humans.
Okay.
But where it's different is for person one on the chromosome that they received from their mother, we can see that they have this AGCT. It's repeated 4 times. So when we analyze and measure this, which we'll go over in the later lectures, we're going to measure it and we're going to be able to say, person one on that chromosome, it's the AGCT is repeated 4 times. And on the other allele for person one, it's repeated 6 times. So when we write out this person's profile, like if this was amplifying D5S818 or something, okay, their pair of numbers for that locus will be 4, 6.
Gotcha.
Okay. Does that make sense? So I'm just trying to visualize like what the DNA looks like. So
Okay.
you know where that 4, 6 comes from. You know where that 6, 7 comes from for person number 2. And if somebody is the same, you can see where the 5, 5 comes from. Okay.
Yeah, that makes more
It's
sense.
not, yeah, it's not that like a 5, 5, they don't just have one chromosome. No, it's that their mother's a 5 and their father's a 5.
Okay, got it. Thank you.
Okay, you're welcome. That's a good question. Are there any other questions as I struggle to use this laser pointer? All right, what I'm going to do is I'm going to stop the recording right now, and then you guys can hang out and you can ask additional questions as you want. So I'm going to stop.